Okay, that was stem cells as therapies. If we look at stem cells as tools, a lot of work here going on for the use of stem cells as screens for drug development. So very useful, obviously, for toxicology and target or biomarker identification for various programs. These are human cells. They're unlimited in supply. They're well characterized. They're stable, and they can be applied to high throughput platforms. And they may offer, they're most likely to offer better predictive utility compared with animal studies. So obviously at the moment we need both, but here is a means of reducing or eliminating animal studies in due course. And again, iPS cells will remove the need for stem cell lines derived from human embryos. Accompanying this is a growth in enabling technologies, robotics, automation, media, 3D scaffolds, and so on. So growth there in stem cells as tools as well. We look at biomaterials and future scenarios for biomaterials. Most of these are scaffolds, so three-dimensional scaffolds, synthetic or bioscaffolds. There are biomimetic scaffolds being developed and are close to the market now, if not they're in the clinic. Injectable scaffolds such as the one developed by Regenerative in Nottingham. Bioactive fibrin-based and synthetic matrix-based scaffolds that Kuros are developing. Bio-inspired scaffolds. Self-assembling polypeptides is an area that is likely to create a lot of interest and be commercially attractive. Bioresorbable coatings for integration cells with tissue, or cells to integrate the biomaterial into tissue, or to be drug-eluting, and possibly linked to point-of-care testing device or strategies, so that those biomaterials become responsive either directly or through a clinician or patient intervention. I couldn't resist showing you this one, which is mine. So this is a three-dimensional scaffold. This is a synthetic scaffold, but the cells populate this scaffold. So this is a tissue infill, tissue filler. This is the injectable self-assembling scaffold that Nanotope are developing. So this assembles after injection. Polypeptide solution is triggered to form a gel in situ by the change in physiological conditions. And they're applying this particularly on spinal cord injury. And they also claim to allow this to allow the claim that increased rates of angiogenesis result as well. And if you look on Google to put this one in, Paralysed Mice Walk Again, there's a video of what they claim to be the effect of these nanotubes. Future scenarios then. So, as I said earlier, there's a lot of technologies that need to go with regenerative medicine to make it happen. A lot of technologies, so diagnostics, biosensors, bioprocessing, and biomanufacture. Delivery, micromanipulation of the cells to the right place, and the supporting areas of manufacturing, clinical trials, health economics, and so on. So a lot of work that needs to be needs to go on in combination with work in stem cells. So some conclusions then. Regenerative medicine is important. It is an emerging industry, and predicted to be over $100 billion industry by 2030. It will change the cost payment model and require good health economics to show the value to health systems globally. Stem cell therapies are certainly at least 5 years away, more than 5 years away, more likely to be 10 years from being routine. Those bridging technologies, scaffolds, smart matrices, and so on, will be important commercially over the next 5 to 10 years. And stem cells as tools will become more established over the next 5 years plus. Changing demographics and continued expectation will drive the demand for this technology, but the barriers will moderate the progress of regenerative medicine over the next 5 or so years. Thank you very much.